You're here because you want more practice on identifying the variables and groups in controlled experiments. Now, all experiments have only 3 variables, one called the independent variable, another one called the dependent variable, and the last one called a control variable. There are also 2 groups, the experimental group and the control group in the controlled experiments that we're doing. Let's recap again. We have the individual, the independent variable, the dependent variable, and the control variable. We also have the experimental group and the control group. An independent variable is defined as the thing that changes on purpose. The scientist changes this on purpose. It's the thing that does the effect, it does the impact. And in this case in front of us, the thing that is changed on purpose is the amount of light per group. Now we have group A, group B, and group C in front of us. We can see that each is receiving a different amount of light. That is what we see that is changed on purpose before the experiment even starts. And that's key. You have to have an independent variable. In our case, we chose our independent variables. We looked at the resources that help the plant grow. In our experiment, we have light, the amount of daily water, the type of soil, the type of plant, and you chose one of those and you chose how you wanted to change it. That is called your independent variable. Your dependent variable, on the other hand, is the outcome of that change. It is the thing that you're measuring as a result of, let's say, for example, in this case, as a result of the different amounts of light. That could be how tall the plant grows, which is height, how big the plant grows, which is the mass. It could be the color of the plant, the number of its leaves. So think of it simply, it is the outcome of your independent variable. It is the outcome of the change. That is why it's dependent. That is why it's the dependent variable. Everything else stays the same, which is also called our control variable. Anything that is not our independent variable is controlled, which means stays the same for every single group. Group A, Group B, Group C. If you have an extra group, you have 2 extra groups, 3, etc. So the question is, then how do we know which group is in the experimental and which group is in the control group? So your experimental group is essentially the group that is affected by your independent variable. In simpler terms, this means that the group that has whatever change you made, that is your experimental group. So what that looks like, this means that group A and B and C in this example are all in the experimental group. That is because our independent variable is different amounts of light. They each have different amounts of light. And because each one actually does have a different amount of light, making it a part of the experimental group or the group that you're testing for. The control group is the exact opposite of that. Control groups are not affected by the independent variable. It is not affected but by what you chose to manipulate, what you chose to change. In this case, in a controlled experiment, the control group would be a group without any light. No light is not the same as a different amount of light because there isn't light at all. The reason why we have a control group and another reason why we have control variables, those 2 separate things, is to make sure that nothing else except the independent variable is affecting the dependent variable. We want to make sure that it's only the independent variable that's affecting the dependent variable, not nothing else. Let's say, for example, I'm arguing that the plant with the most light is the one that grows most in height in this experiment. And let's say I didn't have control variables and I definitely didn't have a control group. Now, based on these results that I just had, it would say that the one with the least amount of light grew the most. So I would change my hypothesis and conclude, all right, the tomato plant with the least amount of light are the plants that grow the most. Based on my evidence, that would be false. Because one, I didn't account for the different amounts of water that wasn't controlled. So that means it could have been the water that made the one with the least amount of light, the one that grew the most, and not the light. Meaning it was the water that affected the height, not the light. But we can't certainly know because now we have two independent variables, which is why we should only have one independent variable and everything else controlled. Now, why do we have the control group? We have that because if we said what we said, that the plant with the least amount of light is the one that grew the most, if someone comes up and say, what if it's a plant with no life that can grow the most? Now, I didn't have a group with no light. Therefore, I can't provide the evidence that that person's wrong. Therefore, I'm wrong. That's why control group is so important and why control variables are really important. They cover our bases and make sure that it's only and only that one independent variable that is affecting what we're measuring and nothing else. That's so we can have conclusive, foolproof results that A, our independent variable, is the only thing affecting our dependent variable. And whatever relationship they have is the relationship they have and nothing else. And there you have it. If you have any additional questions or want more assistance, please reach out to me as soon as possible. Thank you so much.